Okay, so good afternoon, everyone, and thanks for the introduction. My name is Mallika Iyer, and I'm a fourth year graduate student in Dr. Adam Godzik's lab at SPP Medical Discovery Institute. And today I'll be talking to you about the evolution of SARS-CoV-2. But before I get into that, I wanted to say a little bit first about my evolution. So, my journey into science, and in general, a little bit about my growth. So, I was born in India, born and raised in India in a city called Pune, that's about here, close to Mumbai. And these pictures here are basically a representation of what life in India looks like on a day-to-day basis. Because I figured if you go online, you're probably going to see like the Taj Mahal and all those touristy things, but that doesn't really give you a feel for the country. So hopefully this does that. But what I really wanted to talk about was first my journey into science. So I fell in love with biology when I was in the fourth grade and I learned about the digestive system. It just fascinated me to see how the body was like the most well thought out machine that I had ever heard about. And so I pursued biology further, and it was in college that I was introduced to protein structure. And I fell in love with protein structure. And so that's what I wanted to study further from there. But it was also in college that I realized that traditional lab work is just really not for me. I didn't really enjoy it, and I don't think I was very good at it either. So I was thinking of ways in which I could study protein structure without having to be a traditional lab biologist. And so that is what led me to computational biology or bioinformatics. So since I was transitioning to a different field, I decided to get a master's degree first. And so I went to the University of Colorado in Denver to get a master's degree. And I did my thesis research in Dr. Christopher Miller's bioinformatics lab. And so this is where I learned more about bioinformatics and basically developed as a bioinformatics. But this was also a period that saw a lot of development in me as a person. So while I was at CU Denver, I also worked as a tutor on campus at the Learning Resources Center. And this job, which was one of the best jobs I've ever had, really enabled me to expand my worldview. So I have come from a family where everyone has gone to college, and there's actually been more than one person who has gotten a PhD. And so for me, at every step of the way, I've always had people who could guide me in making my decisions. And so, in some sense, I actually took this for granted. But when I went to CU Denver, it is a commuter university, and there are a lot of non traditional students there. So there are first generation students who are the first in their family to go to college. There are students who are coming back to school after, starting a family or start serving in the military. And so this just really opened my eyes to the different experiences that people can have. And it made me appreciate and respect them even more. And so when I decided to give this talk here today at Dazzle, at first I was really intimidated about talking about diversity, equity, and inclusion, because I thought to myself, like, what do I know about that? I've been really fortunate to see the good side of those things. But then, with some encouragement from the organizers, I realized that this experience really is worth sharing, because the point that I'm trying to make, which is hopefully coming across, is that even though you may not have exposure to some of the struggles that other people have faced, that doesn't have to stop you from learning about them and from keeping an open mind and generally being supportive. And so that is the point that I wanted to make with this. And just continuing on with my story about science, after I got my master's degree, I moved on to the next step, and that is my PhD. And so that is what I'm currently doing in Dr. Adam Godzik's lab at SBP. And this project that I will be talking to you about today is a project that we've done very recently. And it's really been a joint team effort. So SARS-CoV-2, I probably don't need to say very much about it. We all know that it's the virus behind this COVID-19 pandemic. A lot of you probably also know that it belongs to the coronavirus family, and it's a positive sense single-stranded RNA virus. I also just want to mention that it has a really large genome that codes for approximately 29 proteins. And these proteins can be divided into three groups: non-structural, structural, and accessory proteins. Now, like most RNA viruses, SARS-CoV-2 is constantly acquiring mutations and it's evolving. So it's already divided into several clades that you can see in this evolutionary tree here. And these clades have distinct geographic and time distributions. So if you look at this graph on the bottom, you see that at different times of the pandemic, different clades have been the dominant clade. But what is hard to understand is whether the mutations that the virus has acquired are actually changing its phenotype in any way. And so in our lab, that's kind of what we're trying to understand. We want to understand how this virus is evolving, and specifically which regions of its genome are under evolutionary pressure. Now, for this virus, we already have a vaccine, but this isn't the first time that we've seen an outbreak of a coronavirus, and it probably won't be the last time either. And so it's still worth it to try and understand more about this virus. And so by studying its evolution, what we hope to gain are insights into the functions of different regions of its genome so that we can identify good drug and antibody targets for the future. And we also think that this will enable the detection of variants with enhanced pathogenicity. But I'll get back to this point towards the end of the talk. So like I said before, our lab is a structural bioinformatics lab, amongst other bioinformatics. But what we wanted to do in this project was to see if we can use protein structures to gain additional insights into the evolution of this virus and what these mutations are doing. So if we look at the sequence here, we see that it has these three mutations. And at the sequence level, they may appear to be pretty far apart. But if we map them onto the protein structure, we can see that they actually get localized to the protein-protein interaction interface. And so this allows us to start making hypotheses about what the mutations could be doing. And so to study this, our lab has created a tool called the Coronavirus 3D Server. And so to create the tool, we combined sequence data from the CNCB and from GISAID with protein structure information from the protein data bank. And so this enables us to view these mutations in the context of protein 3D structure. Now, as I mentioned before, we want to try and identify the regions of the genome that are under selective pressure. And when we talk about selection pressure, we can have either positive or negative selection. So let's say we have a mutation and it gives the virus an advantage that improves its viability. This mutation would be retained in the population of the virus. And so over time, a region that is under positive selection will get overmutated. Whereas on the other hand, if a mutation is disadvantageous to the virus, then it will eventually drop out of the population. And over time, a region that is under negative selection will be undermutated. And so by looking at the distribution of mutations, we can figure out which regions are under positive or negative selection. And usually this is done by calculating the DN to DS ratio. That is the ratio of the number of non-synonymous mutations to the number of synonymous mutations. And so if this ratio is greater than 1, we say there's positive selection. If it's equal to 1, we say there is neutral evolution. And if it's less than 1, we'd say that that region is under negative selection. However, if you look at the distribution of mutations in this virus, what we see is that the variation in the frequency of synonymous mutations is much less than that of missense mutations. So in this graph, we're looking at the number of mutations per 100 nucleotides along the length of the genome. And as we can see, the blue line for synonymous mutations is a lot flatter than the red line. And so this allows us to use a simplified approach that's based on looking at the frequency of missense mutations alone. And so what we do is to use the binomial test. And our null hypothesis is that missense mutations are randomly distributed along the genome, which would be the case if there were neutral evolution. And so the alternative hypothesis is that they're non-randomly distributed, and a region that is overmutated in missense mutations is undergoing positive selection, while a region that is undermutated is under negative selection. And so we use the binomial test. So first we looked at the distribution of mutations in individual proteins. And what we found is that there are 9 proteins that are undergoing, that are undermutated and so are under negative selection. So this is that list of 9 proteins that are significantly undermutated. And if you look at their functions, you'll see that they all have, most of them at least, have a function that makes them essential for the viral life cycle. So for example, you have proteins that participate in the replication of the RNA, or you have proteins that are involved in processing the RNA. In the case of the spike protein, it enables binding to the host cell and entry into the host cell. And so this is basically what we would expect to see, since negative selection would be seen in the case of proteins that have essential functions. On the other hand, we have 10 proteins that are overmutated. And when we look at their functions, we see that most of them are involved in interacting with the host in some way that gives the virus an advantage over the host. So we see that there are some proteins that basically disrupt IFN signaling, enabling immune evasion. Or for example, in the case of NSP1, it inhibits host translation. And so the trend we're seeing is that these overmutated proteins have some type of interaction going on with the host. Now, besides looking at individual proteins, we also wanted to look at domains within proteins, since domains often have distinct evolutionary histories and functions. And so the way we identified domains was both by looking through the literature, but also by mapping the sequences of these proteins to the PDB, because domains can often be crystallized independently. And so if we look at the PDB, we can identify the regions that have been crystallized independently that could represent domains. And so again, what we see here is that even within an individual protein, the different domains can have different mutation frequencies that would be reflective of their functions. So this example here is of NSP3 that has multiple different domains. And we see that these two are undermutated. These two are overmutated, while these two are undermutated. And again, we see that the ones which are undermutated seem to be essential for replication, while the ones that are overmutated have some function related to interacting with the host. Now, the last thing that we looked at was the distribution of mutations within protein structures. So basically, we compared the mutation frequency of buried residues with the mutation frequency of surface residues. And here again, we see a trend that we were expecting to see, which is that buried residues are undermutated while exposed residues are overmutated. And interestingly, residues that form interaction interfaces have an intermediate mutation frequency. So this can be explained by the fact that buried residues, which are at the core of the protein, are tightly packed, and mutating them would probably lead to the protein unfolding. And so they're undermutated. And on the flip side, the exposed residues are overmutated. And the interaction interfaces clearly have functional importance, which could explain their slightly lower mutation frequency when compared to the rest of surface residues. So now, what can we do with these results? How do we put it all together? So we think that we can use this to enable early detection of potentially of variants that could potentially alter the course of the pandemic. So I said that regions under negative selection are undermutated. However, this doesn't mean that they can't get mutated at all. And so if a mutation does occur in such a region, it will provide some significant advantage to the virus in order for it to persist in the population. And so what that means is that we can monitor such regions for potentially dangerous mutations. And a good example of this is this new variant that has recently arisen in the UK. So it has 3 mutations in the spike protein. And these 3 mutations raise 3 red flags. The first is that they change the physical chemical properties of the residues that they're affecting. The second is that they fall in an undermutated region. So if we look at the mutation frequency of the spike RBD, we see that it is significantly undermutated. And the third is that these residues fall on interfaces. So they fall on the interface between the RBD and ACE2, which is the host protein that RBD binds to. And they also fall on the interface between the RBD and various antibodies, like the Regeneron antibody. And so, of course, this then leads us to think, could they potentially be altering binding? And that is something that requires further study and experimental evidence for. But the idea here is that by monitoring regions like this, we could identify variants such as this one that could be having an effect on the phenotype of the virus. And so just to conclude, we first see that the mutation frequency varies along the genome, and proteins and domains that are undermutated are typically essential for the viral life cycle, while those that are overmutated give the virus an advantage over the host. We also see that the mutation frequency varies based on solvent exposure. So buried residues are undermutated, exposed residues are overmutated, and interfacial residues have an intermediate frequency. And so we can use this to identify regions that would be good candidates for inhibitors. So undermutated regions would have a harder time developing resistance mutations, and they're also essential for the viral life cycle, making them good candidates for inhibitors. And as I just mentioned, we can also use this to inform surveillance strategies and by monitoring these regions for potentially dangerous variants. And so with that, I would just like to thank everyone in my lab. Most of the results that I've shown here today have gone into this paper that is currently in review. And it was really a team effort. I'd also like to thank our collaborators, our funding sources, and of course, both SBP and UC Riverside. And with that, I'm happy to take any questions.